Hi everyone, we are going to see about gene therapy. Coming to the introduction of gene therapy, it is the addition of a functional gene in order to correct an inherited defect in an affected individual. Generally, this genetic defect is occurred by means of mutation of a gene. Due to mutation, there is an alter in the activity of an enzyme. For example, consider A gives B. C is the enzyme which is involved in the reaction of conversion of A substrate to that of B end product. In the first case, if the activity of that enzyme is hyper, it makes or it converts all the A substrate into B. If the substrate is an needed constituents, what happened due to the deficiency of that needed constituents, the immune system will be affected. And in the second case, if the enzyme activity is low, thereby the A conversion into that of B is very minimum. If it is a removal of toxic substance, in that case A is a toxic substance which should be converted into B and it should be eliminated from the body. In this case of enzyme activity decrease, this A starts to be accumulated in the body and thereby it will lead to fatal diseases. Coming to the technological aspects of gene therapy, there are 5 major steps are involved before gene therapy is done. The first thing is identification of the normal and the mutated gene. We have to see whether the gene is a normal or mutated. If it is normal, no problem. If it is mutated, we have to go for gene therapy. The second step is generation of a mini gene. Generally, the gene is a larger one. It's not possible for us to transfer into any carrier molecule and thereby we have to construct a mini gene. After the construction, the third step is we are in need of development of a gene delivery system. For the transfer of this mini gene into the recipient, what happen? We have to create a gene delivery system or carrier model. And after the formation of this one, we have to go for the fourth step, which is a regulation of expression of the introduced gene. We have to regulate everything. And finally, preclinical trials is needed. Here 3 different types of preclinical trials are there. We have to do all the things and later only we have to implement into human system. Coming to the types of gene therapy, there are 2 important types or forms of gene therapy. One is somatic cell gene therapy and the other thing is germ cell gene therapy. The first thing is somatic cell gene therapy, which is considered to be an ex vivo addition of gene to isolated cell. The second one is in vivo direct delivery of the corrected gene into the tissue. Under this one, we also have antisense introduction into the target cell of a nucleic acid sequence that is complementary to all or portion of a specific mRNA. Thank you.